I'm Morgan Apolonio. I'm a rising junior at the University of California, Berkeley.
Awesome. Now, would you like to give us a little bit of introduction to the fantastic work that you've been doing in the Gore Lab?
Yeah, so I've been working with the Gore Lab this summer to explore the effect of long non-coding RNA expression on vitamin E levels in maize. And so we know maize is a staple crop. However, it's kind of low in its vitamin E activity, which is really essential for us because at the moment, 80% of the world's population is getting suboptimal amounts, which puts us at an increased risk for cancer, heart disease, and a bunch of other age-related diseases. So we're looking to increase vitamin E activity in maize. And so using GWAS, which is kind of sort of looking at genome-wide population study, seeing which base pairs are significant, they've identified 52 unique QTLs, which are loci. Essentially, they're like, this is really important for vitamin E activity. So we were hoping to resolve some of those by combining TWAS, which has also been done, and they're able to resolve some more of those. But this TWAS didn't use long non-coding RNA, which doesn't encode any proteins, and it's longer than 200 nucleotides. And we specifically looked at intergenic link RNA because it's easier to identify because there's no open reading frames. So when we did that, we kind of sort of ran into a problem where like a whole bunch of our link RNA was expressed in like less than 100 genotypes when we have like 550. So that made a huge problem where it would skew the results of our TWAS. So we decided to only use link RNA expressed in more than 10% of our population, which is like right here. However, we also noted that link RNAs as a whole is expressed on very low levels. With this graph, like you can see it's usually like 15,000 to 2,000 range, and we expect that to be more to gain some significance compared to coding genes. And so when we conducted TWAS, we were able to look at several different chemicals that are significant for vitamin E activity. So one of those is alpha tocopherol. And so we found 3 link RNAs that are significant, which was really nice. And then for delta tocopherol as well, we found another link RNA that was significant. And then we kind of sort of mapped these back to their relative chemical expression. We saw the differences in expression I was talking about earlier, where this one is kind of sort of lowly expressed, which tells us, oh, this one may not be kind of sort of as important, something you want to focus on, but this one has a lot of expression since there's not a lot of values on 0. And so that was nice. And then we combined our significant link RNAs from TWAS. We looked for SNPs that also fell within these link RNA to make things easier on ourselves. And that was done using Fisher's combined test. So this isn't exactly what Fisher's combined test looks like, but it's kind of sort of a representation of it where you take the adjusted p-values from TWAS and the adjusted p-values from GWAS and you combine them. So everything here wasn't significant on kind of sort of both ends, but the ones up here were really significant for both. And so those were located in this link RNA. That's a lot of numbers. So I won't say the entire thing, but that was really nice for us to see and also see it across multiple chemicals important for vitamin E activity. So when we kind of sort of did everything, we were able to identify 13 new link RNA that are connected to the previous identified QTL. So specifically, we have these 5, 2 of which have been kind of sort of close to next to resolved genes. So these 2 genes over here, POR1 and VTE1, we know those are significant in vitamin E activity. And then the remaining 3 we have here are really important for future analysis to see if we can use those to resolve the remaining QTL.
Sounds like you were very busy this summer. Is there one part of this project that really stuck with you and that you're really excited about, or one future direction that you're really excited to see the results of?
I'm really excited to see, I guess, how these link RNA play out, because a lot of the major genes for vitamin E activity have already been identified, like VT1 and POR1. So we have a lot of the smaller ones. So I'm interested in seeing like how those would actually affect vitamin E activity if we were to do like a knockout or do RNA interference with those.
Wow, that sounds like really exciting. Is that something you'd like to pursue after your undergrad or in future career plans?
A little bit, maybe on the RNA interference. And this was all computational work, actually. So we got our genotypes for NCBI, so we didn't do any lab work at all, but I really like doing lab work. So that would be fun, but I don't know, maybe it's not so much on the computational side.
Oh, well, thank you so much, Morgan, for introducing us to your project and for teaching us a little bit about long non-coding RNA. It sounded really interesting, and hope you have a great time at the poster session.
Thank you.
Bye.